Okay, so hello everybody. This is a podcast version of the molecular genetics lecture on eukaryotic expression vectors and epitope tagging. Hopefully this will be a useful revision aid for those of you who wish to revisit this lecture. So today we're going to be talking a little bit about the expression constructs, the plasmids, which we use when working with eukaryotic cells, mammalian cells, higher systems than the kind of things that you've been learning about already on the module, which have been exclusively restricted to bacteria and E. coli. So it's important that you realise that you can't use the same kinds of plasmids to express human genes, for example, in human cells as you use to express different genes in bacterial cells, in E. coli. So today we're going to be talking about some of these different cloning gene expression systems that exist, how we actually introduce plasmids and constructs for the expression of foreign DNA into eukaryotic cells. So it's slightly different again to how you introduce plasmid DNA into bacterial cells. I'll give you some examples of a eukaryotic expression plasmid and point out some of the important characteristics of the plasmids. You'll notice that a lot of the characteristics are similar to those that exist in the bacterial expression vectors, but there are one or two important differences. And then we're going to talk a little bit, in quite a lot of detail really, about different promoters which are used for expressing human genes. So as we're going to cover later on in the module, as I'm sure you're already aware, gene expression in humans in higher systems is far more complex than gene expression in bacterial systems. So you're going to have some interesting lectures from Dr. Ross later in the module about the control of gene expression in bacteria and E. coli via the lac-operon, tryptophan-operon, etc. But however complex those systems might seem, they're relatively straightforward compared to the regulatory mechanisms which exist when controlling expression of human genes. And for that reason, we need to have a little bit more of a complex system available to us, a regulatable system available to us when we express human genes in human cells. And we'll talk why we need that and how that works later on in the lecture. Then we're going to talk a little bit about the concept of epitope tags. So this is a really useful aspect of recombinant DNA technology, which is not only utilised in eukaryotic expression, it's used in expressing things in bacteria as well. But I don't think you'll have encountered this as yet on the module, and we're going to talk a little bit today about what these things are, the green fluorescent protein tag in particular. And we'll finish the lecture by introducing you to another type of expression system which we can use to express human genes in human cells, And this is based on an insect virus called baculovirus. This is a really powerful system for expressing a gene of interest in large amounts in a laboratory situation. And then I'll finish by summarising the main points. So with regards to what you need to take away from this lecture, it's important that you understand the different types of promoters that are used to express mammalian genes, and be aware what the advantages and disadvantages of each of those promoters are. So we're going to talk about a regulatable promoter called the TET on TET-off system. It's relatively complex how that works, but it's important that if you don't understand it from the lecture material, that you revisit that in the core textbooks and to ensure that you do understand how that system works, because it's a really important aspect actually of eukaryotic gene expression, this concept of regulatable control. You need to be able to identify appropriate transfection and expression strategies, be they transient, permanent, or constitutive or inducible. So constitutive or inducible refers again to the type of promoter that is expressing the gene you're interested in. Transient or permanent transfection refers to how you actually go about getting that expression plasmid, that expression construct into the mammalian cell in the first place, which strategy you use. We'll talk about those two different strategies in a moment. You need to understand about the use of epitope tags, particularly GFP. You need to have an appreciation of what GFP is, obviously, but also why it's used, how powerful a tool it actually can be, both for basic scientific research, but also in the clinic from a clinical point of view, and with regards to translational medicine and clinical medicine. And finally, you need to understand how the baculovirus expression system works. Again, this is a complex system when you first encounter it, but you need to persevere with that and make sure you understand how that system works in order to fully meet the learning objectives of this lecture. So there's 4 things that you need to make sure that you're capable of being confident that you can explain in order to meet the learning objectives of the lecture. So to begin with then, we'll talk a little bit about just some basic concepts, just to give you a bit of distance, I guess, between the two different areas of bacterial, prokaryotic and eukaryotic molecular biology. It's important that you're able to confidently deal with these two different aspects without getting confused and merging the two things together. So I often see a lot of exam answers where students confuse aspects of bacterial gene expression with aspects of eukaryotic gene expression. You need to understand that the two systems share a lot of similarities, but they have a lot of important differences. So you need to be confident when discussing either or how things exactly are working, and not to confuse matters, particularly when you're writing essay answers, which you're going to be doing more and more, obviously, now you're in year 2 and next year in year 3. So the first thing to realise is that for the routine cloning of eukaryotic DNA, in other words, the manipulation of the DNA itself via restriction enzymes, the sticking of DNA together via DNA ligase, etc., is still all carried out within bacteria, within E. coli. For the simple reason that at this point, when we're actually just manipulating the DNA itself, E. coli cannot be beaten with regards to how easy it is to work with, how safe it is to work with, and how good it is as a system for within which we can manipulate DNA. However, it's equally important that you realise that from a biomedical science perspective, what we're actually most interested in is looking at the function of human genes. So we can't really do that, we can't do that at all really, by simply expressing human genes in bacterial cells. In order to appreciate how a human gene functions in a human cell, we need to express a human gene in a human cell and then determine the effects of that gene. Moreover, it's not enough to actually just express a gene that you're interested in a human cell and try and work out what it does. You need to express the gene at a physiologically relevant level. So that's particularly important because, as I'm sure you're aware now, different genes are expressed at different levels in different cells actually within the human body. So not all cells express all the same genes all of the time, and moreover not all cells that are expressing a gene express that gene at the same level, in the same amount. And it's that tight, intricate, eloquent control of gene expression that enables you to be as complex as you are. There's not a vast amount of human genes, there's only 25,000 genes. It's nothing really if you think about how complex a human body is. There are mechanisms which exist which increase that number, and then there are several, well, as far as we know at the minute, several layers of regulation. There's likely to be many more that we're still discovering, and indeed we'll touch upon that later on in the module when we talk about regulatory RNA. So there's layer upon layer of complex regulation of how these things are expressed. So if you want to do beneficial, physiologically relevant research on gene expression, you need to be able to control the gene expression of the actual gene you're working with to quite a tight level in order to actually believe that the information that you're looking at, the data that you're gathering, is actually relevant. So until recently, really, expression of mammalian genes was solely from plasmid vectors that integrated into the genome or were transiently transfected and existed for a few cell divisions using viral replication systems as a third option. So this is really the three ways that you could go with regards to expressing your gene of interest in a mammalian cell. You could express it on a plasmid that you integrated into the genome itself, and if you integrated the plasmid, it's called a stable transfectant. You could just transfect the plasmid which expresses your human gene into the cell you're interested in, and the plasmid would be taken up. It will be retained for several cell divisions, but eventually it will be lost because it's not integrated into the genome. Every time the nuclear material divides, there's a chance that one half of that material won't inherit those plasmids if they're not in the right region of the nucleus, and therefore they'll be lost from that cell, that daughter cell. So over several cell divisions, plasmid number dwindles and eventually you lose expression. And finally, people also use viruses for actually replicating gene expression in human cells. And we can talk a little bit about how that works later on as well, briefly. So to just go through how transient transfection and stable insertion of plasmid DNA works. When we talk about the transient insertion of DNA, we're talking about the transient transfection of a DNA plasmid. So the introduction of a piece of DNA a DNA vector expressing a gene of interest in mammalian cells is called DNA transfection. So that's different to the term which is used to explain the uptake of plasmid DNA by bacteria, which is transformation. Yeah, so don't get the two confused. So in mammalian cells, when we talk about getting plasmid DNA into a cell, we talk about transfecting the DNA. It's a transfection. Transient transfection, where we're just interested in expressing the gene for a few cell division cycles, is normally performed under non-selective conditions. No antibiotics. So slightly different to that is stable transfection. So this is where you insert the DNA plasmid as a two-stage process. So the first part is the same as the transient transfection. You just get the DNA into the cell. However, what you then look for is a particular integration event into the genome. So this is where the plasmid DNA recombines with the actual genome of the cell in which it's been inserted. And obviously once it's inserted into the genome, then it is faithfully inherited with every cell division cycle. So every time the cell divides and divides its genome, it also divides the DNA from the plasmid vector, and that gets inherited for, in theory at least, unlimited cell divisions. We identify cells which have undergone this recombination, this integration event, via antibiotic selection. So for example, if your plasmid which has been integrated contains a antibiotic resistance marker for neomycin or geneticin, then cells which have had the cells which have undergone the integration event will be resistant to those antibiotics. Cells which haven't undergone that integration event will not be resistant to those antibiotics and will die. So it's a straightforward antibiotic selection process. So just to recap, transient transfection, you just get the plasmid DNA into the cell. Your gene's expressed for a few cell divisions, but eventually the plasmid is lost, and obviously gene expression ceases. Stable transfection, much more difficult to do. You're looking for an integration event as well as uptake of the DNA, but once you have that integration event, in theory, your gene will be expressed indefinitely in that cell line. So how do you actually get the DNA into the cells in the first place? Well, there are three possible or commonly used techniques for getting DNA, plasmid DNA, into human cells, mammalian cells. These are, to begin with, electroporation. So similar again to bacteria. Electroporation is a very efficient way of introducing DNA to cells. Effectively, brief pulses of electricity generate tiny pores in the plasma membrane of the cells through which the DNA can pass. It works well. It doesn't work well for all cell types because it's quite a harsh technique, a harsh protocol. It damages the cells extensively, so you have a lot of cell death accompanied with the transfection of the DNA. If you're working with a cell line which is particularly tricky to grow and to culture in vitro, electroporation can sometimes just lead to all the cells dying during the process, and you're not actually recovering any of the cells back at all. So you need to take into account the cell type you're working with. Perhaps the most commonly used technique, simply because it's the easiest to carry out, is the use of liposomes. And as I've given an example here in brackets of a common commercially available liposome that's used, called lipofectamine 2000. So what happens here is the liposomes coat the plasmid DNA with a lipid bilayer, which allows the binding and fusion of these DNA liposome complexes to the cell membrane. Some of them have a spermidine in, which has got a really strong positive charge linked to the lipid, and that helps to actually bind to the plasmid DNA which is being transfected. So once these lipid bilayer DNA complexes bind to the plasma membrane, then endocytosis occurs, carries these DNA lipid complexes into the cell and eventually into the nucleus where gene expression occurs. So this is a very common technique. There's been finally a research project, students who have just finished now have been using this compound in the lab for the last 4 weeks to transfect plasmids into human cells as one of my projects, and it works well. They've had some good results. So finally, you've got calcium phosphate DNA precipitate. So this is kind of the old school way of doing it. It's dirt cheap because you can just make the calcium phosphate up yourself in the lab. Effectively, the precipitates that form between calcium phosphate and DNA bind to the surface of the cell and are internalised via endocytosis. It's not as efficient as liposomal transfection, slightly harsher to the cells, but it is cheap. So if you work for somebody who's a bit of a skinflint in science research, then they'll have you trying to transfect your cells with calcium phosphate, and then after a few weeks when it doesn't work, they might eventually put their hand in the pocket and buy some lipofectamine 2000. But these things are important to realise in regards to actual real life research. Lipofectamine 2000 is expensive. 200 microliters, so a fifth of a milliliter, costs about £800, and will last you maybe a month in the lab. So it's an expensive reagent to use, but it does work well for getting the DNA into the cells. So transient transfection then. Effectively, you mix your DNA with, let's say, lipofectamine 2000, and you add that complex to the tissue culture flask within which the cells are growing to introduce a plasmid construct to the cells. These plasmid lipid constructs are taken up by the cells, and eventually, after around 24 hours, you can start to detect gene expression. As we've said, there's no actual antibiotic selection here, but it's not the same situation as what would occur in E. coli, for example, if you tried to transform a plasmid into E. coli and not then select for that plasmid during cell growth. So what you've got to realise is if you transform 10 billion E. coli cells and 1% of those become actually transformed with the plasmid DNA, so you've got several million that have been transformed with the plasmid DNA, and then you continue to grow those cells over the next 16 hours overnight, and you come in the next morning and you want to try and isolate the plasmid DNA that's been taken up by the bacteria. It's not going to work because the doubling time of E. coli is so short, the cells will have increased in number so much that the actual transformants, even though they were fairly plentiful to begin with, have just been diluted out by the vastly greater number of cells which have divided over the night, course of the evening. It's not the same for human cells. The cell generation time is much longer, 24 hours for a cell line which I work with frequently. So if you transfect DNA into these cells, the actual cell number will only increase twofold over a 24-hour period. So if you transfect with 50-60% efficiency, you're still going to have 30 or 40% of the total cells present in the flask containing the plasmid 24 hours later. So this is why we can get away with not using antibiotic selection when we do these kinds of experiments in human cells, just because the cell generation time is much longer. It's very easy to do transient transfection, really. You're not going to see all the cells expressing your gene of interest, but you will see a large proportion of them. Often it's useful to do control experiments where you express a reporter gene in addition to the gene you're interested in. So this is a gene which is easily visualized so that you know that the transfection and expression is working. The best example is green fluorescent protein or other fluorescent proteins. We'll talk about what GFP is later on. But essentially it lets you just quickly visualize whether or not the gene's being expressed in those cells, so you know whether or not the transfection's worked and the gene expression's actually underway in those cell lines. So transient transfection is generally used for preliminary or confirmatory experiments, quick results. It's important to realise that usually it's not physiologically relevant. So when we're transiently transfecting these genes on plasmids into mammalian cells, we're looking at just a very quick high expression of a gene that we're interested in. It's not at a physiologically relevant level. It's not going to be maintained over a number of generations. So it's like quick look-see experiments. If you like, let's look and see what's going on here, and then if we think it's interesting, then we can try and do this in a more measured, careful way down the line. Stable transfection, then you're introducing plasmid constructs into an immortal cell line, or very early on into primary cells, and select after 5 to 24 hours again. Just to point out what the difference is between these two terms in case you've not encountered them, an immortal cell line is what we work with in tissue culture. So an immortalized cell line is a human cell line or a mammalian cell line which will divide indefinitely in culture, at least in theory. And the reason it will divide indefinitely is it's been transformed. Now don't get confused here. I appreciate that there is a chance for confusion when discussing these terms, transformed with relation to activation of oncogenes and transformed with regards to transformation of DNA into bacteria. When I talk about an immortal cell line that's been transformed, I'm talking about a mammalian cell line which has got oncogenes activated. In other words, it's become cancerous. So it will carry on dividing irrespective of how many times it has divided already. A primary cell line is just something that you get from a patient. So if you were working on breast cancer, you may have several immortalized breast cancer cell lines which you can grow indefinitely. But the best clinical model actually for a cell line would be to cut a tumour out of someone's breast, scrape a bit of the tumour into a dish with some growth media and see if you can cause it to grow and divide in tissue culture. Now that's very difficult to do. Primary cell lines aren't easy to culture. They're very tricky to transfect, but they are better model systems for disease. So in either way, what you do in a stable transfection is you get the plasmid DNA into the cell by a transfection, say with lipofectamine again, and then you're selecting for this integration event. The thing to realise is most cells will die. So you transfect, you add the antibiotic to try and select for cells which have undergone the integration event. Most of the cells, 99.9% of them, won't have undergone the event. So the vast majority of cells will die. You're looking for a needle in a haystack a lot of the time, and for that reason it's difficult and time consuming. What you actually do is you need to identify individual clones, so a single cell that's undergone that event. And then when you've identified that cell, you kind of isolate it in the tissue culture flask by placing a ring over that cell and allowing it to grow for several more weeks. It can require several attempts, it takes a long time, uses a lot of reagents. So for those reasons, it's only really used when constructing very important cell lines with defined properties that you're going to work with over a long period of time during the research project. So you wouldn't try and generate a stable cell line, for example, if you just wanted to have a quick look at what a gene did in the cell, or quickly to confirm something that you'd seen elsewhere. You'd only go to the effort of making one of these things if you were going to generate a reagent, a cell line that expressed a gene that you were interested in, and you were going to use that cell line expressing that gene for numerous other experiments over the next year or two, just because of the amount of effort required to generate them. So the final thing then that we can use to express genes in human cells is virus vectors. So there can be certain cell lines, primary cell lines included, which are incredibly difficult to transfect. It's just really hard to actually get the DNA into the cell in the first place. So when that occurs, we have to turn to nature really and take advantage of something that really is unmatched with regards to getting DNA into cells, and that's the virus. So all viruses are concerned with are 1, getting into cells, infecting cells, and then 2, expressing their genes so that they can make more virus. They're the only 2 things that viruses care about. Everything else, all the unfortunate side effects of disease, etc., aren't intentional. It isn't personal. HIV doesn't go out to kill people, Ebola, etc. All they're bothered about is getting into a cell, expressing the genes, making more virus, and getting out of the cell. Fortunately, getting out of the cell invariably means killing it. However, we can kind of turn the tables on the virus a little bit and engineer viruses so that we can use them as a means of expressing genes in human cells. And most viruses have been trialled with regards to this aspect, including viruses like HIV and Ebola. I can imagine a lot of people not being very happy if they were being told that perhaps we were trying to express genes in their body via the Ebola virus. You might have a few people running out of clinics, etc. But believe it or not, they are in clinical trials, a lot of these viruses. And certainly these viruses are useful for expressing genes in human cells, where otherwise we wouldn't be able to because of the limitations of transfection. The most commonly used viruses for this are adenovirus, lentivirus, herpes viruses. You're going to learn a little bit more about the use of viruses in expressing genes in tissue culture, etc. next year on medical genetics. So I don't want to go into much detail about it now. You're welcome to do some wider reading if you like. But just so that you realise that in situations where cell lines are very difficult to transfect using conventional methods, it is possible to engineer a virus to express a gene you're interested in, and then use that virus's innate ability to infect the cells and express genes as a means of getting around that difficulty. Yeah, good. By the way, this is the HIV-1 virus, just this image here. So the eukaryotic plasmids. We've talked a lot about getting plasmid DNA into human cells and the different methods and techniques we use for that, and the different types of DNA transfection that we turn towards depending on what we're trying to do. But what are these eukaryotic expression plasmids like then? How do they look compared to the prokaryotic variation? Well, I just want to take you through this plasmid here, pcDNA 3.1. It's kind of like the PUC19 or the PBR 322 of the mammalian world. So this is like your bog standard simple expression construct. Kind of contains most of the major elements you'll see in all eukaryotic expression constructs. So just to take you through each of these regions, you'll notice some of them are similar from what you've seen in bacteria. So you have a multiple cloning site, which is here, which contains unique enzyme sites which you can use for introducing genes of interest, which have particular cohesive ends. So for example, if you wanted to introduce a gene which you had engineered to have a 5' sticky end of Echo R1 and a 3' sticky end of ZO1, you would digest this plasmid with Echo R1 and ZO1, and it would create sticky ends for those two enzymes, which your sticky ends on your gene would be able to bind to, and then you could use DNA ligase to make the recombinant vector, etc. The same kind of things that Dr. Ross has been talking about in previous lectures. So multiple cloning sites, one thing. Just upstream of that, you'll see this PCMV. So this is a promoter region of the eukaryotic expression plasmid. It's a strong eukaryotic promoter. It's actually a promoter from the cytomegalovirus. So again, we use viruses in eukaryotic biology, because if you're looking to express a gene at a very high level and you want a promoter that can do that robustly and quickly and simply, then again you turn to the things which are most simple and best at doing just that, expressing genes at high levels, which are viruses. So we take the promoter from the cytomegalovirus and we use that in our gene expression constructs, because we know that the viral genes are just all expressed at a really high level whenever the viruses are infecting the cells. Another important thing that we have here is this PA. So this is a polyadenylation signal. So I hope you've encountered before. I think we mentioned it in human genetics last year. We're certainly going to be talking about it later in this module. The concept that when gene expression occurs in human cells, RNA polymerase II copies an mRNA from the DNA template, and then at the end of that mRNA, once transcription has ended, numerous adenine residues are added to form what's known as this polyadenylated tail on the mRNA. That's important for gene expression in eukaryotic systems, in human systems in particular, the poly A tail. So when we express genes in humans, we need to have a plasmid which is capable of adding such a poly A tail, and that's what this region is here in the plasmid, this polyadenylation signal. Remember, we've talked about how whilst we're expressing our genes in human cells, when we go about actually making the recombinant plasmid, when we do all the nuts and bolts of actually recombining this plasmid here with the gene we're interested in, we're going to do all that recombination, that ligation and selection within bacteria still. So for that reason we need a bacterial origin of replication here, because the plasmid needs to be able to replicate in E. coli, otherwise we can't make the actual recombinant plasmid we want, which then gets expressed in human cells. Yeah? So similarly, we also need a prokaryotic antibiotic resistance marker here, ampicillin, and we also have a eukaryotic resistance marker here, neomycin. So this will allow us to select for the plasmid in E. coli when we're trying to engineer and clone our gene. This will allow us to select for the plasmid in a human cell after we've transfected it. For example, if we wanted to try and isolate stable transfectants, bare integration events. It's also important to realise that some contain epitope tags, and we're going to talk about what those are in a short while. There's also mammalian origin of replication. That's this SV40 origin here that allows the plasmid DNA to replicate in mammalian cells. This is an origin of replication from another virus, a different one this time, SV40, which is a polyoma virus. So again, we want the simplest mammalian origin of replication we can find. So let's get one from a virus. Viruses can replicate in human cells, in mammalian cells. Let's get their origin of replication. It's going to be the shortest, the smallest, in a lot of ways the most efficient to use. So that's what a eukaryotic expression construct looks like. You need to be aware of the promoter in particular, the need for polyadenylation signal, and this concept that you also need both prokaryotic and eukaryotic resistance markers and origins of replication. So when we first started looking at developing expression vectors to express genes we were interested in, people just thought, let's just express a gene. If we can do that, then that's great. If we can just express a gene we're interested in human cells, it's a great start. So people went for viral promoters such as the SV40 early promoter, the cytomegalovirus promoter, the immediate early promoter from Roux sarcoma virus, all these really potent, strong promoters. So if they were a curry, for example, these promoters would be like a FAL, yeah? Very effective, but, not very subtle. So the problem with expressing genes from incredibly strong promoters is that what you actually achieve when you get the plasmid DNAs into the cell is a form of expression known as constitutive expression. In other words, the gene that you're interested in is just always expressed, and it's always expressed at an incredibly high level. Now, it's not difficult to appreciate, I don't think, that such expression levels are probably artificial compared to what's going on in the real world, i.e. the human cell or cell within a body. And when you're trying to do experiments to inform you of how genes are affecting biology or a particular system or an individual, expressing genes at artificially high levels can obviously lead to problems. For example, what if you're expressing the gene that induces apoptosis when overexpressed? You're never going to be able to study that gene if you're expressing it at a really high level, because it's just going to kill all the cells in which it's expressed. Yeah? You should remember apoptosis is programmed cell death. I'll just point that out for those of you who've forgotten everything from year one. So the most important thing is that this kind of constitutive expression is probably not consistent with what's going on in vivo in the body. So what we actually need is, in addition to getting the cells into the plasmid DNA into the cells, etc., and expressing it, is we need a way of really having tight regulatable control of that expression. So we need regulatable promoters. So the regulatable system I want to tell you about is this TET-off gene expression system. So this is probably the most commonly used really system at the moment in research labs. And what it does is it allows a precise control of gene expression via the addition or removal of a compound from the growth media of the mammalian cells, tetracycline, or what's actually used is this analogue of tetracycline, doxycycline. One of the reasons it's used is that it's slightly less toxic to the cells. It's also cheaper, which is obviously a factor. What this system allows you to do, the beauty of this system, firstly is that it's quantitative, so you can add different amounts of these things and see different changes to expression level, as I'll show you later on. And it's also reversible, so you can turn on expression, but equally you can turn it off again. So you can give like pulses of gene expression throughout cell growth at different points in the cell cycle. Lots of options there for the scientists who design experiments. And importantly, it's also possible to deliver these things orally to transgenic animals. So if you're trying to create a mouse which controls gene expression, which has the expression of a particular gene controlled by this system, it can feed the animal these things and turn on that gene expression or turn it off. So the link here at the bottom is actually a link to a company's website which gives you more details about how this system works. So a lot of the time with some of the material we're going to be talking about, particularly the technical material procedures like PCR, real-time PCR, these expression systems, some of the best sources for actually extra reading are company web pages. So the companies that sell these sorts of reagents to scientists, as the manuals often have quite nice introductory sections explaining how they work. So in addition to your core textbooks for the module, you could also look on the internet at these kind of company websites and get extra reading from those also. So how does it work then? Well, the system's based upon the E. coli tetracycline repressor protein. So I wanted to try and avoid bacteriology in my lectures and just stick to the eukaryotic stuff. You have enough bacteriology on this module already, so I'm trying to big up the mammalian eukaryotic side of things, but we can't get away from the fact that bacteriology and prokaryotic biology are incredibly useful in regards to providing tools which we can utilise to control gene expression even in mammalian systems. And this is exemplified nicely in the TET-off system. So it uses this tetracycline repressor protein. How does it actually work? Well, I'll tell you the difference between the TET-on system and the TET-off system. There's not much of a difference, but there's one important difference in a second. But what you need to realise is that both these systems, irrespective of which one you go for, use this, at least in this particular incarnation of this technology, the PTRE tight vector. So the example I'm giving you here is the Clontech kit. Obviously different companies sell different named kits depending on what they've patented. They all work or utilise a very similar technology. It's just companies have give their different spin on it. So if you want to look at different examples of TET-off, TET-on gene expression, then there'll be lots of examples you can find via PubMed and via the internet from different companies that sell different versions of this kit. If you want to just focus on what I'm going to tell you today, that's fine. This is a good example of how this technology works. So this PTR type vector then, what does it do? Well, first obviously it contains a gene of interest which you'll clone into the multiple cloning site here. So this could be any gene you're interested in, say a gene you think might cause cancer, whatever. And this gene, importantly, is under the control of this minimal CMV promoter here. So it's a CMV promoter, but it's at a crawl. We're at coma level, not file level now, of gene expression. So the promoter's been engineered so that it barely works. Yeah, it's almost no activity at all. And then upstream of that, like minimal CMV promoter, we have this TET response element here. So this TET response element is a region of DNA to which the tetracycline repressor protein would bind normally within its native host of E. coli. And then we've got other things like resistance markers, poly-A signals, so we can polyadenylate our mRNA of our gene we're interested in. But the important business end of the PTR type plasmid is here. You've got your gene, it's under the control of a CMV promoter that is minimal, that's barely working, and that promoter's just downstream of this TET response element here, to which the tetracycline repressor will bind. So how does it all work? So how does it actually work then? Well, it's actually very clever. So in addition to this PTRE plasmid, which contains your gene under control of this weak CMV promoter, which is just downstream of this TRE element, there's a second plasmid which is also present, which expresses a specially engineered version of the TET repressor protein. So this TET repressor protein is a protein that binds to that TRE element. Now normally when the TET repressor protein binds to the TRE element in E. coli, it will repress gene expression. Obviously that's the name, where the name comes from. However, in this instance, this TET repressor protein has been changed so that it's associated with several copies of this viral transcriptional activator protein VP16. So that's a big word. You don't need to worry too much if you don't get that at first go. What it does, this VP16, is it actually converts the repressor protein into a transcriptional transactivator. Yeah, another big word. What it means is, by adding this VP16 protein or several copies of it to the repressor stops repressing gene expression and it starts activating gene expression. And it does this via the activation of the minimal CMV promoter. So that's why it's called a transcriptional transactivator. Yeah? The trans refers to the fact that when the repressor protein binds to the TRE element, it doesn't itself induce gene expression. What it does is it induces gene expression by activating that minimal CMV promoter. So rather, it basically really ramps up the potency of that minimal CMV promoter, so we start seeing gene expression. Importantly, and this is how the entire thing works, the binding of the TET repressor protein to the TRE element is controlled by the level of tetracycline or doxycycline that's present in the actual media, the growth media of the cells. So it's quite difficult to explain verbally that, so let's have a look at a figure and hopefully that'll help clarify what I've just been saying. So let's start with the TET off system here. Let's start with the TET on system, because I think conceptually it's a little easier to realise what's going on. So this is the PTRE plasmid here. If you look at this region on the bottom, this is kind of just the PTRE plasmid that's been stretched out so it's linear rather than in a circle. And you can see that we've got this PTRE element here, we've got the minimal CMV promoter, and then someone's inserted a gene of interest just downstream of the minimal CMV promoter here. Now what we have here is the TET repressor protein, this blue circle, to which we've got several copies of this VP16 protein, this viral transactivator protein. Now what actually happens is this TET repressor protein only binds to the TET response element when doxycycline is present in the growth media. So when you add doxycycline to the growth media. So what happens is doxycycline binds to the TET repressor protein, and that causes a conformational change in the protein which allows it to associate with the TET response element. As soon as it associates with the TET response element, by virtue of these transcriptional transactivators here, these VP16 proteins, this kind of turns on and activates CMV, and then CMV turns on gene expression. Yeah? So in the TET on system, adding tetracycline turns on expression. And that's because a transactivator only binds to the TET response element when it's associated with doxycycline, tetracycline. In the TET off system, it's exactly the same, but here the transactivator and the VP16s are always bound to the TET response element. So gene expression's always turned on, and it's only when you add doxycycline to the growth media that will bind to the transactivator protein, causing it then to dissociate from the promoter and turn off gene expression. So in the off system, adding tetracycline turns off expression. Yeah. So hopefully that's reasonably clear. If not, you need to just have a look over these figures again, listen to this audio again, look at some textbooks. It's important you understand how this system works. It's not that complex. The key thing is realising that you have a TET repressor protein that's been changed so that it can still recognise this response element, but upon recognising it, it no longer turns off expression, it turns on expression by virtue of its association with these viral transcriptional activator proteins, these VP16 proteins. So what does this look like then if you use this expression system in the lab? We've got some data here. So the graph opposite basically shows you that the system's how sensitive it is, and that it's regulatable in nature. So the key concept is that you can add a little bit of doxycycline and you can turn a little bit of expression on. If you add more doxycycline, you can turn more expression on. So what we've got here is, we haven't got an on-off switch here for a light. We've got a dimmer switch now. So we're not talking about on, off, like a binary switch. We're talking about being able to just turn it up a little bit, then turn it down, depending on how much or doxycycline we add or take away from the media. So you can see here, this is just the company lauding how much better their system is than the leading competitor, I guess. So if you look at the TET-on advanced system, which is what I've just described to you, obviously no doxycycline in the media, no gene expression. This is just a reporter scale for gene expression here on the left, referring to luciferase, relative light units. So luciferase is the firefly gene, which causes fireflies to glow. You can clone that gene, put it under the control of a promoter, and you've got a very easy way of seeing how high gene expression is, because you just measure the amount of light given off via this luciferase protein. So here, when there's no doxycycline present, we don't get any expression. 0.1 nanograms per mil, very little. 1 nanogram per mil, a little bit of expression, still pretty much nothing. 10 nanograms per mil, doxycycline, suddenly we're getting quite a lot of luciferase expression. 100 nanograms per mil, even more gene expression of luciferase. So you've got this inducible, regulatable aspect to gene expression. This is a Western blot, which you're going to hear about in the next lecture, exactly what Western blots are. It's a way of looking at protein expression. So here we're looking at the amount of protein present, and the amount of luciferase protein actually present, rather than just the amount of light that protein is given off. In the TET-on system, no doxycycline, absolutely no luciferase protein expression at all. We add doxycycline, lots and lots of luciferase protein detected by Western blot. The beta-actin here is a control for this experiment. So beta-actin is a housekeeping gene, housekeeping protein. Every cell expresses it, forms the actin cytoskeleton, and we can see that in both lanes here the amount of beta-actin is similar. So this tells us that we're not seeing luciferase here because there's no protein in that sample, because there's plenty of beta-actin there. We're not seeing luciferase because there's no actual gene expression, because there's no doxycycline to turn on gene expression in the system. So I hope that shows how, one, how TET-on, TET-off works, two, some data exemplifying how it can be used to control very tightly gene expression, but also in a regulatable fashion as well. So just a quick recap, because there's been quite a lot of information there. So some essentials about expressing genes in eukaryotic cells. You need a plasmid that contains a eukaryotic promoter, such as a CMV promoter, and amongst other things, such as origins of replication for eukaryotic cells, etc. Generally, we introduce DNA via transfection. So remember, the introduction of DNA into eukaryotic cells is called DNA transfection, and we do that via liposomes or electroporation. And this transfection can either be transient in nature, which is easy to do, or you can try and generate stable transfectants, which is more time consuming and difficult. And you should be appreciative now that regulatable promoters are a very important part of modern molecular biology research, because they allow you to generate situations where gene expression is physiologically relevant. And the best example of that really is a TET-on TET-off system, which uses a specially engineered version of the TET repressor protein to control gene expression. So to move on then, I want to talk for a little bit about epitope tags. So what we've got to now is the point where we've managed to engineer our plasmid. Dr. Ross has talked about how you do that, the restriction enzymes, etc. We've engineered a human gene that we're interested in. into a eukaryotic expression construct. We've done all that in E. coli, and then we've purified that recombinant plasmid DNA, and we've transfected that eukaryotic plasmid DNA into a human cell line using lipofectamine 2000, it's a liposomal reagent. And what we want to do now, we've seen that we can express it, but what we want to do now is we want to study its function. We want to see what it actually does, that gene. So the fact that we can make it in the just make the protein or get the cell to make our gene of interest doesn't really tell us anything about what the gene actually does. So one of the actual quickest ways of seeing or trying to get a handle on how genes function in the cell is to, one of the quickest ways is to visualize where that gene's actually expressed. Sorry, let me rephrase that. Not where the gene's expressed, but where the protein product of that gene resides within the cell. So a very simple example would be a transcription factor. So if your gene of interest turned out to be a transcription factor, in other words it controlled gene expression via binding to promoters, you would expect that that gene would be found in the nucleus of cells, because that's where gene expression occurs, obviously. A quick way of confirming that would be to utilise epitope tags, because epitope tags allow you very easily and readily to detect proteins via microscopy or via Western blotting. And by far the easiest epitope tag to work with in this respect is an epitope tag called green fluorescent protein, or GFP. So the guy who discovered GFP back in the 90s, Osamu Shimomura, was awarded the Nobel Prize for Chemistry in 2008 for the recognition of just how important that discovery and this actual reporter gene GFP has been to modern medical research. He actually isolated it from jellyfish, so Aquaria. So I'm sure you've all seen wildlife programs on the telly where you have jellyfish fluorescing, etc. Those beautiful colours you see that the jellyfish can produce are a consequence of expression of this green fluorescent protein, or a variation of it at least. And it was this Japanese scientist who realised that, well, if you could clone that protein that the jellyfish used to create that light, it would be a very useful reporter tool in biology. Okay, so how do we actually use GFP in the lab then? Well, what we do is we make use of these so-called GFP expression constructs. So these are plasmids which look very similar to pcDNA 3.1 we talked about earlier on. The only difference being, just downstream of the promoter, we have this EGFP gene present. So this is a gene which will encode the GFP protein that fluoresces green. So how you actually go about creating a GFP how you actually go about attaching GFP to your gene is thus. You effectively introduce your gene directly downstream of the GFP gene here, in such a way that when RNA polymerase 2 transcribes GFP, it gets to the end, and rather than stopping, it continues to transcribe your gene that you've inserted. And when it gets to the end of your gene, then it stops. And by doing that, it makes the GFP gene, and then directly attached to it, it makes your gene of interest to create what's called a fusion protein, or a chimeric protein. Now, obviously, there's one important thing that you need to make sure when you're doing an experiment like this, and that is that the GFP gene actually lacks a stop codon. So obviously, the wild-type version of the GFP gene, the last 3 nucleotides in that gene will be a termination codon to tell RNA polymerase 2 to stop. To tell the ribosome to stop translating the protein, and that's the end, and that's a fully mature protein made. We need to remove that termination codon from the GFP gene so that when the ribosome starts translating the GFP protein, when it gets to the end of the GFP, it'll immediately continue translation with the beginning of your gene of interest. So that stop codon from GFP is removed. And what that means is that you create a gene which is comprised of the GFP protein and your gene stuck together, fused together. So what use of the GFP tag proteins? Well, there's a few different examples here of how we can actually use GFP. Before we show some of these videos, I'll apologise if I have to faff around a little bit to get them to play. If they don't play properly, obviously you can look at the actual PowerPoint slides that are just on Blackboard normally outside of this podcast, and they should work on there. This is a bit of a disclaimer in case this podcast technology falls down at this point. What we've got here on the left is actually this is a live image. So the other thing to say about GFP is it allows live imaging of cells. So we can express the GFP protein in a live cell or indeed in a live animal and track what proteins are doing, which is obviously a really powerful thing to be able to do, as exemplified here, where we've got actually cells which are known to be metastatic, and we can see the cells actually, metastatic cells moving around the blood and lymph system of this mouse. We're able to track those metastatic cells as they actually move around inside the body. So you can see that the cells are glowing green here, and we can see them moving around the blood vessels. There's other blood cells in there as well, obviously, but they're not expressing the GFP protein, so they're not fluorescing. It's just the metastatic cells which are. And there's a short video here now that I want to show you, or if it's going to play, which is another example of how we can use GFP to track cells in live animals.
Science Central.
This mouse looks normal until you switch to UV light. Where there's no hair, it glows green. With a hairless mouse, it's all green.